laying down when you're reading, all of those things, your body position plays a role in how your memories are forming. There's lots of things that go into forming non-declarative memories. So things that aren't used, obviously, they go in the recycle bin. Emotion can be linked to visceral motor responses, which I think is super cool, and somatic motor responses, right? When you see the love of your life, your heart goes pitter-patter, right? You might blush a little bit. All of those things are visceral motor responses. Those are things that you can't control, right? Visceral things, autonomic things that you can't control. Somatic motor responses might be to, cover up because you're maybe a little embarrassed, or maybe you start playing with your hair because you want to look good, right? Things like that. You're gonna do things as almost like a reflex when you're around someone that you like, or in response to emotion, right? You hear something really upsetting, and so what do? Right? Those somatic responses, your body's physical response to those things. Our feelings are very subjective, though, and this is where psychology and anatomy kind of meld a little bit in this gray area, because feelings aren't really something you can define. It's not something scientists can quantify. When this area of the brain lights up, we call it happiness. And when this area of the brain lights up, we call it anger. Because in some cases, it's the opposite. In some cases, it's the exact same area of the brain that lights up in happiness and anger. So it gets a little tricky, with all of these wiring things. A part of the brain called the amygdala, which is, again, part of that limbic system, gets a lot of input from these lower regions, like the parts of the brain stem. And so this is how come sometimes when you're really angry or physically sad, you feel sick, right? You feel like you're going to throw up, or you feel like your heart's racing. It's because of this visceral response. Those emotions can literally affect how your body responds. There's a connection between what you're feeling and thinking and what your body is actually doing. It's really cool, that mind-body connection. So let's get back to some more concrete things, although biopsychology is a super cool field, I think. Let's talk about protections of the brain. We can do an organizer for this for sure. So there's 3 major protective measures that we have of our central nervous system. One's called the meninges. One is our cerebral spinal fluid, and one is the blood-brain barrier. So our craniomeninges, they actually continue down and surround the spinal cord. They are pieces of connective tissue that surround our brain and spinal cord. So they're lined up superficial to deep, and so we'll start with superficial. It's called the dura mater. And then we have the arachnoid matter, and then we have the pia matter. These are all the meninges. So some structures that are included in this are something called the epidural space. The epidural space is between the bone and the dura mater. Then there's something called the subdural space, which is between the dura mater and the arachnoid mater. And then we have the subarachnoid space, which is between the arachnoid matter and the pia mater. So this is superficial to deep. So the pia mater is literally sitting right on top of the brain. It's like the saran wrap of the brain. This is all made of dense, irregular connective tissue. Let's talk about the dura mater first. The dura mater actually has 2 layers. So it has an outer fibrous layer. Sometimes it's called the periosteal layer, because it's literally fused with the periosteum of the skull bones. So if you were to pull off the cranium, right, to look at the brain, and you looked inside the bone, the cranial bones, you would see the dura mater stuck to the bone. The dura mater is very fibrous, and it sticks to the bone. So the purpose of the dura mater is to kind of create this barrier between the bones and the brain. And so the periosteal layer does that. It fuses directly to the bone, keeping that together. It's super durable. The meningeal layer is the inner layer. Inner layer called the meningeal layer. That meningeal layer just sits right on top of the arachnoid matter. The dural sinuses are between the meningeal layer and the arachnoid matter. So this is where something called the dural sinuses are, which the dural sinuses is where the venous blood flow of the brain will be. So we'll put them kind of in here. Dural sinuses, venous blood. You'll talk about these next semester when you do cerebral circulation. It's basically little pools of blood surrounding your brain. So the dural folds, these dural sinuses embedded in the dural folds, they kind of invaginate or kind of go into the lateral hemisphere, right? Our longitudinal hemisphere, that big fissure that divides the two hemispheres. It dips down into that. It dips down in between the cerebrum and the cerebellum, so it forms these little extra kind of nips and tucks around the brain. It embeds and enfolds all around the brain to keep it all together. We call them dural folds. It's just folds of the dura mater. The subdural space, and then we get to the arachnoid matter. The arachnoid matter has arachnoid, they're called trabeculae. I don't think your textbook calls them anything, but that's okay. You don't have to know it if it's not in your book. The trabeculae literally look like spider webs. So the reason this is called the arachnoid matter is because this almost looks like cobwebs surrounding your brain. So you'll see this in the picture, but oh yeah, it does call arachnoid trabeculae. Go me. Okay, there's also arachnoid granulations as part of this. The arachnoid granulations are where the CSF is reabsorbed. And we'll look at that, obviously, when we look at cerebrospinal fluid in just a minute. And then you have the subarachnoid space. And then below that is the pia mater. This is the deepest. This is right on top of the brain. This holds blood vessels. I'm just going to abbreviate BV. Holds BVs onto the brain. Right? It keeps the brain tissue together. It's literally like saran wrap going around the entire brain. It goes into every single sulcus, every single fissure, dips into every single groove on the surface of the brain. Fits it like a glove. So here you see some of the structures that I was talking about. This is kind of a weird image, but you have the cranial bone. Oops. Deep to the cranial bone is your dura mater. Below your dura mater is your arachnoid mater. Below your arachnoid mater is the pia mater, right? This is showing you those dural sinuses, right? So this is our dura mater. The sinuses are cavities. So it's basically like a tube that's being made from these membranes, right? And this is where blood pools. So you can see this blue line that's representing venous blood. You'll see them in lab on the models when we look at the brains. This is a close-up view. You can see those arachnoid trabeculae, right? Whoops. Those little spider webs. It looks like a cobweb, right? And the pia mater literally goes into every single groove. The pia mater is covering every single. If you had a brain in front of you, you would not be able to separate the pia mater from the brain. In fact, in lab, we will dissect the brains, and I'll challenge you to pull the pia mater off, and it's very, very hard. You can in some parts, but for the most part, that pia mater it's like Velcroed on. It doesn't come off. The ones that we have in lab don't have arachnoid or dura mater. It's just a brain. Whoops. All right, let's talk about the ventricle. So we can't talk about cerebrospinal fluid until we identify that our brain has cavities in it, openings, holes, if you will, chambers that allow this CSF to circulate. And so this kind of goes back to the different types of neuroglial cells. We're going to talk about a few different types of neuroglial cells here. So if you forgot those, they're back to haunt you. Ependymal cells line these ventricles, and ependymal cells have cilia. Remember those? Those are the ones with cilia. And so those cilia just kind of wave back and forth and allow the CSF to circulate through the ventricles. So there are literally chambers and holes in your brain where the CSF accumulates. CSF is cerebral spinal fluid. And that's our other layer of protection. Right? The ependymal cells with their cilia help circulate and pass the cerebral spinal fluid through the ventricles. So there's 2 lateral ventricles. You don't have to know the horns of the ventricle. I thought I deleted that. The horns of the ventricle you don't have to know. You just have to know there's 2 of them. There's a right and left lateral ventricle. There's something called the third ventricle. The third ventricle is in the thalamus. And then the fourth ventricle is right near the cerebellum. Right? And so the CSF is made in a structure called the choroid plexus. Let's use CSF circulation. This is going to be a flow chart. So CSF is made at the choroid plexus in the ventricle. So all of these ventricles, all of these openings that we just named, the right and left lateral ventricle, the third ventricle, the fourth ventricle, they all have structures on them with specialized ependymal cells that form something called the choroid plexus. So we'll start at, we'll just say, because this is a cycle, it could really start anywhere, but we'll just say we're going to start at the lateral ventricle. So I'm just going to call this the lateral ventricle here, even though CSF is made in all of the ventricles, but because we're going to draw this as a circuit, we have to start somewhere on the circuit, right? So from the lateral ventricles, CSF flows through the interventricular foramen. Foramen's a fancy word for hole, right? Through the interventricular foramen to the third ventricle. The third ventricle is within the thalamus. Remember the thalamus kind of looks like a lollipop, a little ball on the end of a stick. So inside that ball is empty. It's a ventricle. And there's ependymal cells in there circulating CSF. So the third ventricle in the thalamus. And then from the third ventricle, CSF goes through something called the cerebral aqueduct. And then it goes to the fourth ventricle. From the fourth ventricle, I'm trying to switch pen, our CSF goes through the central canal of the spinal cord. Then once it gets to the end of the spinal cord, it's going to loop up and it's going to now travel in the subarachnoid space. And it gets reabsorbed into the blood at the arachnoid granulations. So that's the whole flow of CSF through the brain and spinal cord. This will make sense if you're having trouble visualizing. It'll make sense when you see a diagram. Go ahead.
What does it say, like, choroid?
Choroid plexus.
Oh, okay.
Yeah, Plexus. It's going to be on the next couple of slides. I got ahead of myself because I just wanted to get it up here since we're running short on time. So this is a picture of the actual ventricles, right? Spaces in the brain, right? So here's our lateral ventricles. You have one on the right and one on the left. Interventricular foramen, third ventricle, cerebral aqueduct, fourth ventricle, central canal. From the central canal, that CSF kind of sweeps up. Remember, the central canal only goes to about here, right, where your lumbar vertebrae start. It's going to circulate up in that subarachnoid space, right, just below the arachnoid matter. And there's little arachnoid granulations, and it'll get reabsorbed and filtered back into the blood. So, what does it do? CSF. We see how it circulates, but it's there to cushion the brain. It acts as like a water balloon barrier right around the brain. It's also helping to remove waste from the brain. So the blood and the brain don't mix. That's the function of the blood-brain barrier. We'll talk about that in a second. So the CSF is kind of like the specialized circulation to the neurons, right? And so it's there to help maintain a constant temperature, constant ion levels, constant glucose levels, right? Constant everything to make sure your brain has what it needs. Right? I think we talked about this a little bit, like it doesn't matter if you're exercising or you're sleeping, your brain always gets a constant volume of blood, right? And that's why, right? It has to maintain stable conditions. Your brain has to be stable there. So the choroid plexus, this is that specialized area lined with ependymal cells. This is what creates your CSF. It does it through special capillaries called fenestrated capillaries. You'll learn about these when you do circulatory system next semester, but they're capillaries that have a little opening or a little hole in them. So you make about 2/3 of a cup of it at any given time, and then the old CSF has to be reabsorbed by those arachnoid granulations. So it circulates, it only does one cycle. It's made, circulates, reabsorbed, right? So it's not returning to where it started, right? But it could start in any of the ventricles. Right, the pathway of it could have started anywhere. The process of this flow is about 5 to 6 hours. So the CSF that you have now is different than the CSF you had this morning when you woke up. Right? It takes 5 to 6 hours to do this whole cycle. It's reabsorbed and then recreated again. So where is it going? Ependymal cells, right? And this is the exact pathway that I just drew over there, just in a little bit more detail here. Whoops. Right, those ependymal cells are what create the CSF. Showing you this whole pathway. The choroid plexus, usually the choroid plexus on models is shown in the lateral ventricles, to our third ventricle, fourth ventricle, down, and then it goes back up in that subarachnoid space where it can get reabsorbed by those arachnoid granulations. Let's talk briefly about the blood-brain barrier. Just have a few minutes left. This is what's separating the blood from the CSF. So this is a chemical barrier. It's a chemical and biological barrier. So it's preventing bacteria that might be in your blood, viruses that might be in your blood, drugs that might be in your blood, right? Anything that's bad that might be in your blood from getting in touch with your brain. That's the whole purpose. It's a protective measure. That's what all of these are, protective measures, right? So I think it says that these are specialized cells that are making up the blood-brain barrier. This is mostly astrocytes, if you remember our neuroglial cells, mostly astrocytes that maintain this. So these endothelial astrocytes, right, they literally form a network. They interlock and overlap each other, right? And they form this barrier, like a brick wall around the brain so that nothing can get through, right? They're going to limit what comes in contact with the brain. There's only a few spots where there's little breaks in this blood-brain barrier, and that's at the choroid plexus, right? The choroid plexus kind of is like a filtering station for the blood. The blood comes up from the heart, goes to these little choroid plexus stations, all the good stuff's taken out, goes into the CSF, forms CSF, right? And then at the arachnoid granulations at the end, all the crap, right, waste products, carbon dioxide, is dumped back into the blood, right? So the blood doesn't actually have to come in contact with the brain. It's all the good stuff, glucose and oxygen, come out here, and then all the waste products, right, carbon dioxide and the other waste products all go back, right? So there's a limited capacity for your brain to come in contact with dangerous things. The barrier is protective, but this can also hinder any medications that need to go to the brain. It makes it very difficult to treat medical conditions involving the brain, right? So if you're on any, those drugs that help you with depression or anxiety or anything like that, they have a very complex chemistry to them. What you know if you've ever been on them, the side effects of them can vary. I mean, isn't one of the side effects of a depressive drug suicidal thoughts? Like, how does that work? Like, isn't that the whole purpose of taking the drug, right? So they're very and it varies from individual to individual because all of our brain chemistries are very different. But the idea is it's only letting the good out and it's allowing for the removal of the waste products at those arachnoid granulations. This is just showing you the gist of how that blood-brain barrier is put together, right? Water, solutes, important things can filter out. Those astrocytes kind of close up those capillaries. Don't let the things out. Prevent them. Only at the choroid plexus. There's a few other parts of your brain that have a break in the blood-brain barrier. We'll look at those later. But that's the gist of it. So we'll stop here with the end of the blood-brain barrier. We'll pick up there on Thursday. Your practical's tomorrow night. And I was saying at the beginning of class, I may use some